Welcome, welcome. This is where we're going to talk about cellular respiration. This is a metabolic pathway that will allow cells to harvest and store chemical energy. So in the center of it all, there's an organelle called mitochondria. Mitochondria will be responsible for ATP synthesis. And we know that ATP is the energy molecule that cells are able to utilize to carry out different biological work. Notice mitochondria have outer membrane, inner membrane that's highly convoluted, and that's where the machinery for ATP synthase is actually embedded. Understand that cellular respiration is the set of metabolic reactions, and these reactions are going to be happening in series of small steps. So a lot of energy is going to be released when we break the bonds between carbon and carbon, carbon and hydrogen. In other words, we're gonna be oxidizing food molecule, in this case glucose, into CO2. So we're converting the energy from one form to the other, chemical to chemical. Notice the inputs of cellular respiration would be glucose molecule and oxygen. This is why we eat and we breathe. And then the waste products are going to be carbon dioxide and water. Behind the scenes, this energy is going to be transferred to form ATP. We have to have ADP molecule attach the inorganic phosphate to it and synthesize a fully charged battery that we call ATP. There are 4 linked biochemical pathways that will make up cellular respiration. Notice the very first step happens outside the mitochondria, so we call that glycolysis. The next step happens inside the mitochondria, we call that pyruvate oxidation. And the next step, number 3, citric acid cycle, also called Krebs cycle, followed by last step, oxidative phosphorylation that includes electron transport chain and chemiosmosis. During these biochemical pathways, we generate some ATP here and here, but the majority of ATP will be generated at this last step, and we call that through the process of oxidative phosphorylation. So now the very first step is called glycolysis. Glycolysis happens inside the cytoplasm because that's where the sugar molecule is transported into first. So if the sugar molecule glucose has 6 carbon atoms, 6C, we will end up having 2 3 carbon compounds, and we're going to call these substances pyruvates. Let's take a look at how these reactions are happening. So notice we have 2 main phases, energy investment phase followed by energy payoff phase. We call this energy investment phase because we're going to be utilizing ATP to get those reactions going. So glucose in the cytoplasm will be recognized by an enzyme called hexokinase. And this enzyme will also recognize ATP. So those 2 substrates will sit in the enzyme's active site, and catalytic reaction is going to be happening. ATP will be hydrolyzed, and the phosphate group will be added to the glucose. So we say we have phosphorylated glucose. So now this phosphorylation at step 1 and also at step 3, notice, except it's carried out by a different enzyme, it's going to trap the glucose in the cytoplasm so that this glucose is destined to continue on in a pathway and split in half and become pyruvate. As you can see here, your glucose molecule has been phosphorylated. In other words, this compound becomes very unstable, and an enzyme at step 4 is going to cleave the compound into 2 parts. So you're going to get 3 carbon compound here and 3 carbon compound here. Notice no carbon has been released in the form of CO2. We just broke a bond to separate this molecule into 2 components. And then each of these compounds will continue to step 6, and you can see here an enzyme is going to remove hydrogen and electrons and donate that to a molecule that we call NAD+, electron acceptor, and NAD+, therefore, becomes reduced, becoming NADH. So this is your carrier, electron carrier molecule. We will continue to synthesize, pick up these NADH molecules along the way so that we can use them later to power the synthesis of ATP. The way we generate ATP here is via the process of substrate level phosphorylation. I like to call it low-key production of ATP. Notice this enzyme at step 7 will remove phosphate group from this compound and add the phosphate to the ADP, and therefore we will have the synthesis of ATP. And then another enzyme is going to do the same. You can see here at step 10, this is where the last phosphate group is removed, added to the ADP, and now we have another ATP produced. So the end product of glycolysis would be 2 3-carbon compounds, and we call them pyruvates. So now understand that we only gained 2 ATPs so far. We invested 2, we produced 4, the net gain is only 2. So which means majority of energy is still trapped within pyruvate. So the next process that's going to happen after glycolysis will be pyruvate oxidation. So notice now we're actually moving into the mitochondria, specifically into the mitochondria matrix. So pyruvate will be transported into the mitochondria matrix, and this is where pyruvate oxidation is going to take place. The bond, notice right here, is going to be cleaved, and carbon dioxide will be given off as waste product. The electrons are going to be released and captured by NAD+. NAD+ will accept 2 electrons and 1 hydrogen and become electron carrier molecule, which we can utilize later in that very last step of cellular respiration, where we actually synthesize lots of ATP. So what we get here from pyruvate oxidation is a compound called acetyl. So this is a 2-carbon compound because we lost 1 carbon in the form of CO2. Notice there is a coenzyme A that's going to join acetyl, and this coenzyme A will also have a sulfur group. This part is actually a derivative of vitamin B, and it will facilitate the transfer of acetyl and bring the acetyl into the next set of reactions that are going to be called Krebs cycle or citric acid cycle reactions. So now that we have acetyl group, we are ready to begin step 3, which will be citric acid cycle, also called Krebs cycle. But before I go over this cycle, I want to go back and revisit exactly what we had here before. I'm going to simplify things here for you. So notice that first in glycolysis we had 1, 2, 3, 4, 5, 6 carbon atoms. After glycolysis, remember we cut this bond right here and we end up having 1, 2, 3, 1, 2, 3. These are your pyruvates. And then in step 2, what happened was we oxidized pyruvate. So it means we cut the bond right here, and we released one of the carbons in the form of CO2, and the same thing right here, CO2. What are we left with? We are left with acetyl, which is a 2-carbon compound. And as I mentioned before, acetyl doesn't come alone into the Krebs cycle. There is a coenzyme A that's going to join the acetyl and deliver acetyl to get those reactions going. So now when the acetyl arrives, which is a 2-carbon compound, it will bind to a pre-existing compound that we call oxaloacetate. So there's 4 carbons in it. So 2 plus 4 will make a 6-carbon compound, and we call that compound citrate. This is why we call the cycle as citric acid cycle. So you might wonder, wait a minute, now we're building molecules? We were supposed to be oxidizing them and breaking them, right? Well, the reason we're going to have all these 8 reactions happen here because we want to maximize the production of NADHs and also another carrier molecule such as FADH2. Remember this, that NADHs and FADH2s are your electron carrier molecules, so we can transfer the energy to the last step of cellular respiration, and that's where we're going to synthesize a lot of ATP. And by the way, one NADH will eventually yield 2.5 ADPs, and then one FADH2 will yield 1.5 ATPs. So we're going to see how this process works. But first, let's go back to the citric acid cycle. So now we established that we produced a 6-carbon compound called citrate. And the process is going to begin. Step 2, notice citrate is turned into its isomer. No carbon is lost. And in step 3, now we do have a loss. We go from 6 carbon compound to a 5. So then you say, where did the carbon go? And the answer is it left in the form of CO2. So now, since these bonds were broken, electrons were released, NAD plus picked up electrons and hydrogen and became reduced into NADH. And I say, cha-ching, we have little coins for our piggy bank. What's the piggy bank? When we're going to cash in? We're going to cash in at the very end of cellular respiration during oxidative phosphorylation. And then we continue to step 4. We go from 5 carbon compound to 4. Again, where did the carbon go? Well, it was released in the form of CO2. So it means we came in here having 2 carbons in the form of acetyl. And then notice we already released those 2 carbons that we had. So it means we have completely oxidized the entire glucose molecule. And every single carbon was released in a form of CO2. So the last few steps that's going to follow, notice, will be there to basically regenerate oxaloacetate, because we want to have that pre-existing compound, so that way when the acetyl comes into the cycle, we can continue the process over and over again. So at step 5, notice we generate some energy molecules. So GTP will be eventually turned into ATP, so we call that substrate level phosphorylation. And then at step 6, notice we are passing the electrons to another electron acceptor, which is FAD. So FAD will pick up 2 hydrogens, 2 electrons, and become reduced into FADH2. So this is another electron carrier molecule that we can synthesize, that we can utilize later. So notice no carbon atoms were lost or released. So it means the bonds that were broken had to be between carbon and hydrogen. So that's the source of hydrogen. And then the last 2 steps, notice here we're going to get one more NADH, but since the cycle turns twice, meaning we have a cycle for each acetyl that came in, so notice we're going to generate 2 NADHs here at this step. So now oxaloacetate has been regenerated. If another acetyl comes in, we can continue with the entire process. All right, and we are finally at step 4. This is what we call oxidative phosphorylation. Oxidative phosphorylation has 2 components. We have electron transport chain and also chemiosmosis. This inner membrane of mitochondria, the one that's highly convoluted, has lots of proteins that are embedded within. So that is your electron transport chain that's going to be involved in passing the electrons all the way to the final electron acceptor, which is going to be oxygen. So now, remember how we had the reaction step 1, which was glycolysis. We made some NADHs there. Step 2 was pyruvate oxidation. We also made some NADHs here. We also had Krebs cycle happening where we generated NADHs, and in addition, we produced a few FADH2s. So all of these molecules were electron carrier molecules, and that's where they're all coming to. So they are arriving at the electron transport chain, and there they will drop off the electrons to be passed down the chain and accepted by oxygen. So remember, oxygen is the final electron acceptor and is going to be pulling those electrons down the chain. So the way we pass the electrons is by a series of redox reactions. So notice NADH will be oxidized, will give up 2 electrons to that first compound here, and then it becomes NAD+, so that means it's the oxidized version of it. And then this compound will send electrons to the next one, and then to the next one, and then finally notice here FADH2 will donate its electrons to the second complex. And this electron journey continues through all the cytochrome complexes until the very end. Notice here, and we're talking about oxygen. So we inhale oxygen. We require oxygen for aerobic respiration. So oxygen will pull those electrons down the chain because it's highly electronegative, and together with hydrogen, they will get reduced. Oxygen, electrons, and hydrogen get reduced into water. This is why we say water is a byproduct of cellular respiration. So this is where the water is actually forming. So now you might wonder, okay, but what is the point of these electrons traveling through the ETC? What is this process coupled to? And the answer is this. When the electrons pass down through the ETC, they activate hydrogen pumps that are embedded within this system. So you can see it one here, and here's another one. So these hydrogen pumps are going to be able to move hydrogen into the intermembrane space, very narrow space between the outer and inner membrane. So the next step would be chemiosmosis. When we generate the gradient of hydrogen in the intermembrane space, the hydrogen is going to be diffusing through ATP synthase, which is not only a channel for the hydrogen to diffuse through, but it's also an enzyme. So it has two domains. It has a channel for the hydrogen to go through, and then it has a catalytic enzymatic domain where the synthesis of ATP will be taking place. Now let's put this all together. So notice right here we have a outer membrane of mitochondria, and this is that. So this is the inner membrane of mitochondria, and electron transport chain is embedded within the membrane. So you can see we have all the complexes there, and hydrogen pumps. All the NADHs and FADH2s that we generated previously will arrive to the ETC and they get oxidized. So they release the electrons into the ETC. And the electrons will be moving from protein to protein to protein along the way, from acceptor to donor through a series of redox reactions until finally they get picked up by oxygen. So oxygen pulls electrons, attaches hydrogen, and then together they are going to become reduced into water. So now as the electrons are traveling through the ETC, the hydrogen pumps are going to be activated. This is where you're going to see hydrogen moving into the intermembrane space in mitochondria. So the high concentration gradient of hydrogen, we're going to call that proton motive force. When the hydrogen has nowhere to go, because remember this is an ion, so you can't go directly through that membrane. No. So it means the only way for them to diffuse out of this tight space is through ATP synthase, which is not only a channel, but it's also an enzyme. So as hydrogen is diffusing down a gradient, they activate this catalytic domain that begins to spin. And therefore, this part of the enzyme will allow for phosphorylation of ADP into ATP. So this process is called chemiosmotic phosphorylation. So this is where we utilize oxygen. We utilize hydrogen gradient, electrochemical gradient, to generate a lot of ATP. So this is why we collected the NADHs and FADH2s along the way to power this machinery and generate ATP. And the last few things that I wanted to add here is just to remind you that we explored sugar metabolism, meaning how we oxidize glucose to generate ATP. So remember that we don't consume just carbohydrates. We also eat proteins, we also eat fats. So those are going to have to be broken down into amino acids. If these are proteins, fats will be broken down into glycerol, fatty acids. And you can see they will be entering these pathways at different positions. And then also keep in mind that not all organic molecules of food are going to be destined to be oxidized to produce the ATP. Now remember, we also have to make carbon skeletons so that you can build your own molecules. So make cells and then tissues and build the entire body. Next, we're going to talk about what happens when there is not enough oxygen present in the cells, meaning what other metabolic pathways will be taking place in those cells, and I will see you then.